Hello and welcome everyone. You are joining us here tonight for our AP Bio Long Free Response Labs and Data review session, and I'm so glad that you guys are here. A lot of you guys have already answered the poll that I put up. Feel free to enter your responses if you're just logging in now, and we'll talk about that in a second. We're going to do introductions and then talk about a little bit what the session is going to be about and then get right into it. So thank you guys for joining us. My name is Sarah Lassiter. I am an AP Biology teacher at Research Triangle High School in North Carolina. You may or may not know of my husband, Patrick Lassiter, who also does some of these reviews on Fiveable. And he does world history. I'm an AP Biology teacher, though, so I'm here tonight to help you guys out reviewing and prepping for this long free response section of the free responses on the AP Biology exam, which is coming up in less than a week. So I hope you guys are feeling ready and prepared. And if you're not quite there yet, hopefully tonight will help you guys out a little bit. If you want some more resources, I know Fiveable has a lot of excellent stuff that you can find on their site, and you can become a member if you want. You can also find some of my resources on my YouTube channel or follow me on Twitter. And all right, so let's go ahead and get started. Please make sure you go ahead and answer the poll if you are interested in sharing some of your information. We're going to be talking specifically about free response questions tonight in AP Biology. But if you have any other general questions or you want me to go over specific things, feel free to answer, ask a question within the Ask a Question section, or talk in the chat a little bit, and you guys can interact with each other and answer each other's questions if you would like as well. So in looking at the poll, it looks like a lot of you guys are nervous about content on the free response section, which we will be reviewing different types of content that may or may not show up on the AP Biology FRQ thing. We are also going to be looking at experimental stuff, a little bit on timing and pacing, and thank you guys for answering the poll. It looks like a lot of you guys have ideas about this. We're going to go over some graphing stuff as well, but mostly we're going to be talking about tips and strategies to do well on the long free response section, and also go through a few practice problems together tonight as well. This review should last about an hour, and we're going to start by doing practices and doing a little strategies before we get into our practice about tips and general guidelines about this FRQ, specifically the long free response section, a little bit of background on it, and then after that, we're going to dive right into some practice. So let's go ahead and get started. All right, so the free response questions on the exam, as some of you may know, this comes in section 2. So you have already done the multiple choice by the time you get to the free response, and you are given a 10-minute reading period at the start. The full free response section is 90 minutes. Now you can go ahead and get started with your free response questions during that 10-minute reading period. There's no one stopping you from getting started, but we really recommend you taking that 10-minute reading period to sort of synthesize all the information that is given to you, and to also go through and plan out your responses. So if you allocate time according to that, you would have 10 minutes for reading and planning. Then your proctor will tell you your reading period is over, and then you can go ahead and start answering your questions with the 80 minutes that are left. The question types that are going to show up on the free response section, there are going to be 2 long ones. And tonight we're specifically focusing on the style and the ways you can best answer those long free response. But a lot of the tips and strategies we're going to go over here can also be applied to the short free responses, which of which there'll be 6 of them, and there are 3 or 4 points available to those. But the long ones are longer. They're broken up into different sections. They are 10 points each, and as we'll talk about later, they're weighted a tiny bit more than the other free response questions on the exam. So some students tend to really stress out about these questions because there's so much. They're so long and there's so much in them, and sometimes they have experiments they may or may not have ever seen before. So we're gonna cover some strategies related to those. If you are allocating your time exactly, breaking each one down, each long question you should plan to spend about 22 minutes on, and then each short one you can maybe spend about 6 minutes on. But again, this may or may not equal out perfectly when you're doing the AP Bio exam. You may go faster on certain ones may take you a little bit longer. But when you're working you're going to want to have to plan your time out appropriately based on what you know and where you're going with your questions. So let's talk about free response questions in general. Again, you got to answer these in blue or black pen. That includes the graphs, so you'll have to have that with you on the day of your exam. You'll probably have a pencil for the first part. You want to put that away for the free response section. College Board approves most graphing calculators these days. It used to be you could only use a calculator with your 4 functions on it. They don't make that happen anymore. You can bring most graphing calculators, but check on the College Board to make sure your calculator works. Of course, you need your brain. You do not need scratch paper. You do not need a pencil, like I already mentioned. And of course, you don't need your phone, but hopefully you've already put that away before you actually go into the exam room. You will also get a formula sheet on the free response question section, just like you will get a formula sheet on the first section of the exam. And we'll go over what this looks like and how you can best use it. You're probably going to need to use your formula sheet on the free response section of the exam, especially if a question like a chi-square formula pops up, and that will be provided for you. So you do not need to go memorizing some of those topics. All right. I saw in the chat, can you bring a laptop to the test? No, unless you have a special accommodation for that as well. All right. So again, if you have any questions that are popping up as I'm going through this information, feel free to put them in the Ask a Question box, and we'll continue to keep going. All right. So free response questions, best ways to prepare. I tell my students this all the time. You can Google and find the released free response question sections from old AP exams pretty easily. So if you just Google straight up AP Bio free response 2013, you can find the questions that were released to most students. Now there's still secure exams that are out there that teachers have access to that you won't have access to, but the College Board is generous in that they have all those past FRQs out there, which you can find and practice with. So if you haven't already, I would recommend sitting down and looking through at least one full FRQ over the weekend before the final exam comes up. It's a great way to practice and sort of get into the mindset of how that exam is going to feel on the day of the test. They also have the old scoring guidelines, which means that you can go and grade yourself after you answer the FRQ practices. So go through, quiz yourself, and that's kind of what we're going to do tonight. We're going to take an old FRQ, we're going to go through, we're going to answer it, and then later on you can go back and score your answers. Of course, I'm going to use scoring guidelines to formulate my answers, as you will be able to compare yours to. But this is a really useful tool that you can use to sort of help frame your studying. So again, you just Google AP Bio FRQ 2013 scoring guidelines, and it should pop up with the full FRQ and all the responses. What's interesting is they'll release the FRQs for this year about 48 hours after the exam comes out. So you are not supposed to talk about the exam after it finishes, but the FRQs are something that you can discuss once those have been released to the general public once you're done. So it's a pretty useful tool, and I recommend using it. One thing is that do not look for FRQs to practice with before 2012. This is when the exam actually underwent a large change. And so there are a lot of FRQs out there that exist before 2012 that are not going to be reflective of what is actually on the exam. So make sure you start around 2013 and go forward. And we'll talk about what is on what has been on the FRQs recently since the redesign, and how those can be helpful to frame your studying. All right, general tips. Plan your time. Like I said, the long ones are supposed to take about 22 minutes, but that's really up to you. If you want to kind of use that as a guideline, if you go way over 22 minutes on the first one, you might want to take a pause and go back and answer some of the other ones that are simpler for you. But again, everybody's different. Everybody's going to take a different amount of time for each question. But I do recommend using the reading period. So plan an order of attack. Even if you don't start with the long FRQs, please make sure you decide when and where you're going to answer them in your period of 80 minutes. So the long FRQs will both exist as the first 2 questions on the AP Biology exam. So those will be the 2 longest questions you'll have to answer. Other ones may have long reading sections or passages or graphs or things you have to interpret, but the questions with the most subsections like A, B, C, D, E, that'll be the first 2 questions on the exam. So that's what I mean when I say the 2 long questions. Those will be question 1 and question 2 on the FRQs. I really recommend starting with the easiest, but again, the long FRQs are worth slightly more than the other questions, so it is good not to abandon those. And I do know students who skip the long FRQs because they feel like they can get more points on the other ones that seem easier. Please don't skip them. It's a really good idea to answer as much as you can and to tackle it even if you're not sure 100% about the correct answer. So again, don't leave questions blank. There are really easy points that are out there. Sometimes you can get a point for just titling a graph or labeling axes on a graph, or you can get a point for circling the right part on a cladogram, or you can get a point for identifying the right part of a diagram that it asked you to. So it's a really good idea to take a stab at a question, even if you're not sure. You're not going to get points off for entering a wrong response. Yeah, so make sure you write something on all questions. Do your best to answer it, even if you have no idea, because you may get lucky and you may get a generous point here and there. There are usually a few easy points available on every single question. So even though you may not get all the points available to you, it is good to just try to respond with something related to something you learned this year or sounds like it reflects one of the science practices that we have to know for AP biology, but do your best to try to get some of those easy points. And be sure to answer what the question is asking. We're going to talk about those command terms like describe or calculate or construct, but make sure that you are really putting your answer in the terms and with the words that the response is asking of you. So underline those command terms. They'll be in bold actually on your FRQ, so you'll be able to see pretty easily what the question wants you to do. If it wants you to construct or like draw something, it'll say construct a graph. Or if it wants you to calculate, it'll bold calculate. If it wants you to describe, it'll bold that word. So it's a good idea to circle those just as a reminder to figure out exactly what that question is asking. So also if it says like list one strategy or list two advantages, list two, but don't do more. And when I say list, you still have to write in complete sentences. We'll talk about format in a second. But generally, it's not a good idea to go over what the exam asks of you. It's also a good idea not to be too wordy as well as you go through the exam. All right, so I'm going to take a pause real quick and take a look at our questions. There's one that has a couple of upvotes. It says, for an FRQ question that involves writing on a provided graph or cladogram, do you have to use pen or can we use pencil? They're wondering because they're not sure how you can fix something on a graph when you write in pen. You do have to write in pen on a drawing when you're making a graph, or if you are writing or circling something on a cladogram. And what that means is that you may want to jot down your response or sketch out an idea for a response on the side. You may use a pencil and then go back and write over the responses in pen. I do advise against this though, because sometimes you run out of time and you have to go back and write over all of your words or all of your graph lines. But really, really try to do your best to do your best answer in pen. If you mess up on a graph, what I tell my students is, especially if you're like doing the scale and all of a sudden you realize you messed up the scale, just do one line with your pen and cross through it. You don't have to like scratch it out and make a black box over it and destroy it. But just draw one line over it and then put your correct answer in the spot where you want. If you make a huge mistake on the graph, again, just make sure you X out the part that you do not want the graders to grade, and then you can write over it. But I would give you the advice is to kind of plan your response and plan what you want to do outside of the response section and the side margins where they tell you to plan your ideas and plan your responses. There's plenty of space for that. But yes, you do have to draw in pen. So that is one important tip. So I'll get back to some of the other questions there in a little bit. We're going to keep going with some of these tips and then we're going to get to practice pretty soon. So thank you guys for hanging in there. All right. Next up, more general tips. So you do have to write in complete sentences. You must write in paragraph form. This means no bullet points, no numbered lists, but that also means that you don't have to write perfect essays. So you don't have to obsess over perfect grammar. There's no deductions for points for grammatical errors or for spelling errors. As long as the readers know what you're trying to say, they will give you points for it. So say you mess up a few letters in photosynthesis. It's not going to be a big deal. If you write something though and you make up a term and you try to pretend it's a word and you make something completely up, they may not give you points for it. But do your best to write understandably and write correctly, but you don't have to worry about, is this a sentence fragment or something if you're rushing for time. So again, avoid those bullet point lists, outlines, and drawings. Those will not be graded unless it tells you to draw in a specific space. And then there, yes, that drawing will be graded or that graph will be graded. You do not need to write introductory or closing paragraphs. You do not need a thesis statement. This is very different from maybe some of your humanities AP exams. You don't get extra points for topic sentences, so you do not need to ramble. So again, don't obsess over perfect grammar or spelling. You don't get points counted off for poor sentence structure, but you do need to try your best to write in complete sentences, and make sure they end in periods, that kind of thing. All right, another way to avoid going overboard with writing, because sometimes you get caught up and you lose time this way, is to not restate the question. So if you notice on our practice today, we'll keep our answers pretty straightforward. You will make clarifying statements to make sure the readers understand what you're saying, but you don't need to go back and restate the entire statement or experiment in order for the graders to understand what you're saying. So those long FRQs, the first two questions with several subsections, they're going to be worth a tiny bit more on when the questions are graded. Not a huge amount, but a tiny bit more. So it's a good idea not to skip them again. So a really important thing that we're going to do tonight and that you should do in your review is to review experimental design. Things like how to set up a good experiment, what a variable is, what types of variables they are, how to write a null hypothesis. And all of these things may show up and probably will show up on your long FRQs. Out of those two long FRQs, one, the College Board guarantees will have a lab or some sort of data that you'll have to interpret. It might be both of them, but at least one will have labs and data. So that is where we're putting our focus on tonight. You can avoid lengthy explanations, so anything like thus or fancy language or whatever. Just make sure you get over it. And then in other words to avoid, remember you are still taking an AP biology exam. You have to be scientific. So anything like molecules loving, like I know it's easy to say like a hydrophilic molecule loves water, but technically they don't have feelings, so try to avoid that. Prove, again, we never prove anything. We only accept or reject our hypotheses, or we support our hypotheses. So don't say prove. And again, speaking in absolutes is never a good idea. So try to keep those words out of your FRQ writing as much as you can. And again, just gut check your responses. If the question's asking about photosynthesis, you probably don't want to reference an animal cell, or vice versa. If it's referencing an animal cell, you probably don't want to start talking about chloroplasts. So think about what you're writing and take a step back and breathe and relax as you're going through these FRQs. All right, so we're getting into some of the specific strategies here. Again, please keep answering each other's questions and asking things within the Ask a Question section. I know a lot of you guys are nervous about the content that's going to show up, but we're going to talk also about some timing and pacing strategies as well as designing experiments in just a second. All right. Oh, and yeah, and I saw in this chat Skylar's comment about how the beginning of the test just throws everybody off because those are the most difficult questions, and it's designed that way. Those two questions, the two long free response questions, are typically the hardest of the exam, or at least they're the most dense, and they're broken down into many different subsections, and they have sometimes several figures, sometimes a lot of data. Most of the time it's going to be experiment you'll probably have never heard of before, and it is designed to sort of throw you off. But that's why that reading period is really important, and that's why it's important also to sort of take a deep breath, practice these long FRQs with experiments and data, and don't get freaked out by them, because they really are not that bad once you start practicing them. So we're gonna get into it right now. All right, so when we get into these math sections of the long free responses, sometimes you're going to be asked to analyze a set of data. Sometimes you're going to be asked to conduct a chi-square test, which is a statistical test that a lot of you guys have studied over this year in AP Biology. Sometimes you're going to be asked to analyze a graph and interpret different values from a graph. But it's really important to use that formula sheet that is provided for you on the exam. You can find out what this looks like by just Googling the AP Biology formula sheet online. It's pretty straightforward, but if there's any formulas on there that you don't know how to use, or have variables or values that you don't understand, practice those now so that when you get to the exam, if you have to use that formula, you're not confused about what they are. If you do any calculations, even though your math may not necessarily be graded, it is a good idea to show work that you have. So if you, for example, are calculating the density of a population within an area, you may want to show how you set up that problem and then put your answer right above it or right below it. So again, show your setup. Good graphing conventions. So I'm kind of skipping around here, but once you get to actually designing a graph, and we'll talk in a second about how likely it is that you're going to have to draw a graph on the AP Biology exam, these are things that you can always generally get points for. So one, title. Never forget to title a graph. If you're making a graph, do it as the first thing. It's always very important. Also make that title specific. Don't just say, for example, there's the question we're going to practice tonight has to do with an experiment with caffeine and bees. You just don't want to leave your title as caffeine bees or bee graph or something. You want to make sure you basically state the experiment. And sometimes there will be a table title within your FRQ. You can use a very similar title as is given in the figures on the FRQ. So don't be afraid to sort of reallocate the information already in the FRQ to your own graph. Axes need to be titled appropriately as well, and they need to have units. So please make sure you identify each axis with the proper units that you are showing on your graph. The scale needs to be appropriately calculated. You also don't want to make a really teeny tiny graph in the space provided. So they will give you a graph section. You won't have to draw graph lines or anything if you do need to draw a graph. And you will have to place your graph appropriately. So you can't have your graph extend off the page. And this is where some students mess up their scale as well. They make their scale too small, and then their graph has to go, or too large, and they have to extend past the graphing section. You don't want to do that. Again, back to that pen thing, if you want to like lightly sketch out your scale before you actually get started drawing your graph, and then go back and write in darker lines on the scale, that might be more effective for you. So just make sure you count the number of lines you have, and then look at the data that you have to represent and see what the most effective way will be to represent that data. Make sure you're using the correct graph type as well. If you are supposed to be representing different categories, you probably don't want to show a line graph. So we'll talk about how to choose the right graph in just a second. But you can get points for choosing just the correct graph, even if your data is all wrong, or vice versa. You know, if your axes and your title are correct and you have the right units, but you graphed incorrectly, you can still get a point for that. So that's why it's a really good idea to take a stab at some of these questions, even if you do not know for sure exactly how you're supposed to do it. If applicable, it's a great idea to draw error bars. We're going to practice those in a little bit as well. Error bars are going to show the margin of error that data falls into. And if those error bars end up crossing each other, and I'm doing this with my hands now, but you'll be able to see it on the graph example that we have. So if there's any overlap in those error bars, that data set is probably not showing a significant difference. And that is a common thing that the AP exam likes you to point out. So keep an eye out for any error bars that might overlap within an experimental data set, and you will be able to see this on a graph. And that is something you should watch out for. If your graphing or if your data set shows any sort of standard error of the mean, so SEM or standard deviation, that's the kind of an alert that you should be drawing error bars. And what you'll do is you'll extend an error bar like a little T hat off the top of your data point. And again, I'm doing this all verbally. I'll show you the example in just a moment. And then extend it below as well. And that will be how you set up your error bars. So we'll get back to that. I know I did not have a visual there to go along with it, but we will review error bars in just a second. Okay, so choosing the appropriate graph for your data. You're going to use a line graph if you're measuring a change in something generally over time. So you're going to represent a gradual change, like let's say you're doing amount of rainfall over several different months. But anytime time shows up as part of your data set, you probably want to be thinking about a line graph. A bar graph is going to be comparing different individuals to each other where there's only one data point. So for example, if you're measuring the density of the density of pollinators, I keep thinking about this bee question, in different environments, then you would want to show the different environments as the different categories and the density as that one value with the bar graphs. Again, think categories, you are comparing different points, you're going to use a bar graph. Very, very frequently, the graph you will have to design on the AP Biology exam will be a bar graph. That is the most common type of graph I have seen. That's not guaranteed. You may have to draw a line graph or a pie graph or a different type of graph, but most likely it is going to be a bar graph if you have to draw one. So that's just in the past, historically, what we have seen on the AP Biology exam. So a pie graph, though, is going to show percentages and components of a whole. So maybe you're looking at the biomass that takes up an environment, or maybe you're looking at the components of a cell after you have run it through a centrifuge and you're looking at the different parts of it. So any of these things could be a graph that you may have to design. But if you have to draw a graph, again, the axes or the graphing paper or the area of which you'll have to draw it will be provided for you. So you won't have to just, freehand lines in an empty space. So that's available to you as well. All right. So remember that the formula sheet is your friend, but make sure when you are going through and looking at labs within a long free response question, you look at the experimental design first. So don't necessarily jump right into the data set. You want to take a look and make sure you understand what is going on within the experimental setup. So typically within a long free response lab question, they'll tell you some complicated, long-winded explanation of an experiment that has been designed. They might give you a data set, they probably will, and they're going to ask you to do several things with that data set. Sometimes it'll be graph the data, sometimes it'll be interpret the data, sometimes you'll be asked to calculate statistical significance for the data, sometimes you'll even have to draw something. But very frequently you will be asked to design a new experiment or to design an experiment to show something. And when you have to do this, you have to make sure you know a couple of important experimental design things right off the top of your head. Now, when you're designing this experiment, you do not have to start from the very beginning of time. I have seen students' responses where they start and they're like, go to the store, go to the laboratory company, purchase seeds, purchase soil. Don't do any of that. You just need to say the bare bones of your experiment. So make sure you start with your variables. What are you changing and what are you measuring? So your independent variable, I am the experimenter, remember I for independent, what am I going to manipulate or change within this experiment? My dependent variable, what is this experiment depending on? And what am I measuring here? So for example, if I'm taking, let's say my acid rain experiment I did with my biology students, a lot of them chose an independent variable as the concentration of acid within or the pH of a solution and the different pH levels of on seed germination. And then their dependent variable would have been the number of days until germination or how much they measured. So how many millimeters the seed actually ended up germinating. That's my dependent variable. And then my independent, again, was the incremental changes that I gave as my treatments to that plant. All right, your control is going to be what you are comparing. It's your comparison group. Sometimes this is just no treatment at all. So very commonly within an experiment, you can just say the control group would be flour with no treatment or whatever. So you can use that phrase too as your control. Very commonly, water is also a control in an experiment if you're doing something with solutions and different concentrations with solutions. So think about common controls that you could throw out there when you're designing experiments. You also need to know how to construct a null hypothesis. Remember, a null hypothesis is the idea that your experiment will have no effect. The changes that you're showing are not going to demonstrate any statistical significance. There's no experimental difference. Now, the way students get confused with this a lot is that the null hypothesis scientists have to construct, but it's typically not what the scientist wants to see in the experiment. It's sort of the opposite of that. But you need to get used to creating and generating null hypotheses and being able to state them. And they can be really simple too. So for example, you could say the treatment has no significant difference from the control. That's a great way to state a null hypothesis. In genetics, you could say if you are stating a null hypothesis for a group, you would say that the data are consistent with the predicted method of inheritance. So whatever we predict, say through complete dominance, that's what we're going to get in this experiment. So the alternative hypothesis, that if there was a difference, would be that the data are not consistent with the predicted method of inheritance. All right, so if we are moving along, I'm going to peek at the chat real quick because I haven't been paying attention into it. People are talking about different types of graphs, rarely scatter plots. So scatter plots are going to show up that as graphs you'll have to interpret. I have not seen any time where a student has had to construct a scatter plot on a recent AP biology exam. All right, so let's move on forward with some practice. Okay, so when we're talking about past exams, so here we go. This is a chart, and I'll leave this up here for a second because we're going to discuss what has shown up. I know a lot of you guys are anxious to predict what's going to show up on the exam, and us teachers are too. This is kind of the general outline of the topics that have shown up from questions 1 and question 2, the 2 long FRQs on past AP Biology exams. So my face is kind of down here in the corner over 2013, but this is the first exam set after the exam redesign, and we'll be looking at one of these tonight, actually. Anything shaded in this darker color here, this is a question where you would have had to construct a graph. So, or construct a cladogram. I forgot to mention that. So cladograms or phylogenetic trees sometimes come up in place of a graph, or sometimes you have to do both. Very frequently, though, the cladogram will be provided for you, and you'll just have to put the organisms on the correct taxa. So you'll have to put them on the right tips of the cladogram. So going through, and if you guys want to look at any of these or practice any of these things, say, for example, you're really concerned about the immune system, you're really concerned about cellular respiration, you can use this chart to go back and find the question that corresponds to that topic and practice it this weekend before the exam. So from 2013, the question topic was on animal behavior-ish. Again, a lot of these questions hit on many different topics. So you're not just going to see animal behavior. Sometimes it's animal behavior and evolution, or animal behavior and molecular genetics. So this is just like a generic summary of these things. But you also had to be able to calculate a chi-square value on that question as well. We're actually going to review part of this FRQ tonight, so hang tight on that one. The question 2 on 2013, another long one, was on photosynthesis and evolution. You did have to interpret a graph on that one, but there was no graph construction. 2014, moving up, there was a trichome density question. It was an experiment, pretty straightforward. You did have to construct a graph there. It was a bar graph with error bars. You also had to construct a phylogenetic tree or your cladogram that year. And the question was sort of on evolution related to the immune system. 2015, question 1 was on it was a long question with a mouse and circadian rhythms and selection. That one was pretty complicated just because it had a lot of figures. You did have to interpret a figure or a graph on that one. You did not have to construct one. And then question 2 was on cellular respiration, and it gave you a lot of the processes within cellular respiration. So that's an interesting interpretation of figures. And you had to calculate energy efficiency in that one, but it was a pretty straightforward math problem. In 2016, question on genetic variation and osmoregulation, molecular genetics, kind of. You did have to construct a graph for that one. I don't remember off the top of my head. I think that was a bar graph as well. Gene expression, population growth, population density, and growth rate were all involved in part 2 or question 2 of 2016. And you did have to calculate population density for that one as well. That was a bacteria question. All right, question number 1 from 2017. This is the one we're going to review tonight and talk about some strategies for it. It was an experimental design question. Very, very interesting, kind of cool question about caffeine and bees. And you had to construct a graph for that particular question. Guess what? It was a bar graph. Question 2 for 2017 was about environmental regulation and germination, and you had to interpret a graph. So even on the same year, you had to construct a graph and interpret graph, but that's very common. There's a lot of graphs that you're going to have to interpret probably on the AP Biology exam, not just in the FRQs too. And then question 1 from 2018, the most recent one that's been released, evolution, molecular genetics, genetic engineering, kind of all wrapped into one. You did have to construct a cladogram. That was the question with a bunch of bear stuff and rats. Kind of interesting. And then question 2, there was some data that you had to interpret, and it was like a cell signaling question. So you had to interpret what was going on in a model or a figure, and there was a question about water balance, also the immune system. So that's a pretty good question. I would recommend visiting any of these if you are curious about any of these topics, and you can go and quiz yourself and review some of these exams. But as you can see from the past 3 years, you've had to either construct a graph or a cladogram. So it is very likely that could come up on the AP Biology exam. Now, I'm only showing you here questions 1 and questions 2 from the old FRQs. I didn't go and show you what was showing up on all the other questions. Question 1 and questions 2 are the long free response questions. The short ones could also have you construct a cladogram, or they could also have you do something small on a graph. They're just not going to be as extensive. So the big graphs that are going to show up are going to be on either question 1 or question 2. But you could also have to draw something or make some sort of notation on something before you end up at the end of the exam. All right. So like I said before, highly likely you will get some sort of graph to construct, not necessarily true, but I'm going to guess it's probably going to show up. And I'm also going to guess that you'll probably have to construct a cladogram at some point as well. And you're probably going to have to make some sort of ideas to how to design an experiment. So those are all things we're going to hit tonight. Actually don't have a cladogram question up, but you guys can access those pretty easily. And we're going to go ahead and start practicing some of these things. So for those of you who joined in late or who are just coming on right now, you can either ask a question in the question box. I'll try to answer those in just a moment and try to get through some of these. And as you are transitioning to our practice, I'm going to add a link here in a moment. Let me move on to our next slide. Here we go. This is a document that we're going to do some long FRQ practice in. So I'm going to walk through what I would write for the responses for each of these and talk through where students make mistakes and some strategies for answering an FRQ like this. Even if you've already seen this FRQ, because it's a great one, your teacher might have shown it to you before, I would recommend sticking with us and following along on the strategies for how to answer this question. So you can go ahead and make a copy of that document. The first one we're going to review is the 2017 AP Biology FRQ question number 1 about caffeine in bees. And then the second one is the 2013 question number 1 about fruit flies in a choice chamber. So again, if you've seen these FRQs before, again, hang tight. There'll still probably be some valuable tips that you're going to get out of these. But if you haven't, hopefully you'll be able to practice them for the first time with us. I also have the link to that document posted as a green button on the Crowdcast, but I will put this in the chat box as well in case you just want to copy the document and follow along. So I'm going to pull that up in a second so you will be able to see my screen. And here we go. All right, so that should link you to the document if you haven't already accessed it. And as you guys are pulling that up, I'm going to answer some of these questions that are popping up in the box. All right. So will there be an FRQ for each big idea? So evolution, molecular biology, ecology, et cetera. So there's 4 main big ideas, and they are kind of all over the place sometimes, but most of the FRQs will hit on, or excuse me, in the exam, they're going to hit on all the big ideas at some point. Some will be more heavily than others, and some will be a combination of several big ideas. So you might see something related to more cell signaling that's also related to evolution, but a lot of the big ideas are linked. So yes, it's a good idea to cover each big idea if you've heard your teacher reference those before, but it's not necessary going to be like big idea 1, big idea 2, big idea 3, big idea 4. So there's that. All right. Is it a good idea to skip around on the FRQ so that you have time to think and answer a bit of everyone at the very least? That is a good strategy. Definitely use your reading period when you get to that part of the exam and sort of plan out your plan of attack. Answer the ones you know off the top of the head first or you feel very comfortable with first, but save time for each of the questions. So yes, skipping around is a good strategy, but don't leave those long free responses for last because you may not get to them and they are really important and there's easy points available to you. Okay, so related, how should we best use our suggested reading time? What are some good methods for going through questions and outlining the response if that's something you should even do? So we're going to talk a little bit of some of those strategies for those questions tonight as we go through those FRQs. One thing I would say is to read through the questions, especially the experiments first, just absorb the information, circle any key terms that you see, circle any of the command terms. So those are those bolded words that say construct or describe or calculate. And that'll trigger your brain to remember that's exactly what you're supposed to be answering. After you go through once with the experimental question, I'm going to want you to think about the figures in the experiment. So take a quick look at all the data tables, all the graphs that are provided, any images, any models, and see if you can take a few seconds to sort of interpret what's going on. Then go back and look at what's being asked of you and maybe jot down in the margins a couple of ideas that are popping into your head. Once you have a quick idea of what the question is asking and anything that really jumps out at you, it's a good idea to move on to the next one and do the same strategy. Read through the full-length question, circle any of the command terms, look at the figures, and do it again. Once you have kind of a general idea of what everyone looks like, then I would go back and maybe order them. Maybe put like a number 1 next to each or create an actual outline, a bullet point list that you can go and check off. But it's a good idea at the very end to, even when you're rushed for time, maybe with the last few minutes of your free response period, this is at the end of the exam, to go back and make sure you haven't skipped anything, especially if you are jumping around. So those are some strategies, but we'll go over a few more as we start to do this practice. All right, so we're moving on to the actual practice FRQs. So hopefully you guys have pulled up that document and made a copy for yourself. You can go ahead and start answering if you would like. I'm going to answer it on my end too, so you can follow along. Please feel free to score yourself, either with the scoring guidelines. I recommend putting those away. You can easily find them online, but don't click on those right away, and make sure you're using the time to practice for yourself. So as you guys are pulling stuff up, if you want to go ahead and start reading through the questions, and I am going to here we go. I'm going to make sure that you guys can see my screen where we will be answering these questions. All right, and I have to pull it up this way. All right, here we go. Hopefully you guys are seeing what I'm seeing in just a moment. It's loaded. All right, okay. So if you want to pull up the actual 2017 FRQ question, you can click on a link. I'm not going to pull it up just to save room on the screen as this is coming, but I'm going to go through some of these pretty quickly. If you want to follow along with the actual College Board page, the link is right here, and hopefully you have your own document to write on so that you can go through and put your answers in and follow along as we do our version. All right. So let me know if there's anything that doesn't come up on the screen or if you can't see anything appropriately, and I'll try to check in as we go through. Okay, so we're going to start with a graphing question. This question is on question 1 of the 2017 scoring guidelines. The first thing we see is a table. So if you're looking at this question with me right now, you're probably going to see that table effect of 0.1 millimolar caffeine in memory on bees. So I would not even look at that table right away. I mean, it's interesting, and there's it's pretty simple and straightforward. I would go straight to the paragraph when I'm reading this question and read about the experiment. I know it's about caffeine in bees, and that's pretty much it. So it talks about in flowering plants, about pollination that leads to the fertilization of an egg and the production of seeds. Some flowers attract pollinators such as bees using visual and chemical clues. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower and use it to make honey for the colony. Nectar contains sugar, but certain plants also produce caffeine in the nectar. So again, all of that leading up to this point was just background information, which is helpful, but it really isn't about the important part of the question. Caffeine is a bitter-tasting compound that can be toxic to insects in high concentrations. To investigate the role of caffeine in nectar, a group of researchers studied the effect 0.1 millimolar of caffeine on bee behavior. The results of an experiment to test the effect of caffeine on bees' memory of a nectar source are shown in Table 1. So that is the first key important thing that we are looking at. You are asked in question number 1A, on the axis provided, construct an appropriate labeled graph to illustrate the effect of caffeine on the probability of bees revisiting a nectar source. So I want you guys to think, if you're sketching this out at home or if you have this up, think really quickly about what type of graph you think this should be based on the information provided. If you think you know, you can either put it in the chat. If you're not on the chat and you don't want to toggle back and forth, that's fine too. I'm not going to draw the graph on the Google Doc because that would take a really long time. Excellent. I'm seeing a lot of you guys adding bar graph. Good. So it is a bar graph. And go ahead and type in, if you would like, the title that you would give this graph if you are constructing it on your own. If you've already peeked at the scoring guidelines, don't go ahead and type that in, but go ahead and type in a title that you would give to this particular bar graph. Remember, that's one of the first things you should do when you are constructing a graph. And as we have that coming up, I'm going to go ahead and add in the graph that I have created. I drew this on my own copy. I printed out the FRQ this afternoon, and I wrote it out. Yeah, probably something really similar to chart title. Excellent. Yeah, that's exactly what you would want to do. So a lot of you guys have great ideas in there. Perfect. Okay, so hopefully you can see the graph that I added. I'm going to make this tiny bit bigger because you can see my messy handwriting way up close. All right, So this is the space that I have provided. I need to work in the space. You guys can see I did it in pen, and you can kind of see like my chicken scratch and dots as I went up the side on my scale. Again, I don't have great handwriting, but try your best to be legible on the actual AP exam. You notice my bars are not perfectly straight. My error bars aren't perfect either. Let's talk a little bit about the construction of this graph before we move on. So scale is pretty straightforward. You just need to calculate and see how you want to divide up the different sections. My axes, on one side I have memory, but then in parentheses, I have the same definition of memory that they use in the table. So average probability of revisiting a nectar source. And then at the bottom, I have treatment, but I made sure to identify the differences between the 10-minute section and then the 24-hour section, and the 2 bars for each. The shading is not necessary. You guys can see where I shaded my graph, unless I didn't want to include control and caffeine labels at the bottom and I wanted to make a key. That would have been fine too. If you make a key, just keep it close to your graph, not way off to the side. And if you do shading for a bar graph, just make sure it's like really clear which one is shaded. If you have more than 2 bars and there are 2 different conditions, you make it very clear, like stripes or I don't know, clear stripes, bubbles, very shaded, but don't go crazy with the shading because that's very difficult to read. Why am I doing this as a bar graph? I'm comparing different categories. So I'm looking at 10 minutes and I'm looking at the 24 hours results. And I'm also making sure I included my error bars. It is important to look at the standard error of the mean that they gave us in the data table. And from there, I'm going to extrapolate, if you guys can see, from the point at which my data stops for the bar and going up for this particular one. This is the control after 10 minutes. So my data point is at 0.72 plus or minus 0.09. So I want to go up what I think is about 0.09 and put the top of my error bar there. And then I want to go down the same amount, 0.09, and put my error bar bottom there. Now, when I look at these, I see that there is overlap between my 2 error bars. So this is basically saying this data point could fluctuate anywhere in between this section, and so could this one. And so what this means is that there's no significant difference in these 2 data points because there is overlap. So you might want to think about that later on. It could come up. All right, I'm going to leave this up here, and I will give you guys access to this document later so you can compare your answers, but we're going to move forward just for time's sake. All right, so when we're talking about the next part of the question, oh, one more thing I forgot to mention. A lot of students on this question ended up getting their label of their axes wrong because they just left treatment down here. They didn't actually differentiate like the categorical data. And so make sure if you have categorical data, you label each of the categories. If you have continuous data, you can just have that one label like memory up here because this is a continuous spectra data. But down here, because it's categorical data, we want to make sure we have each of these categories labeled. All right, hope that makes sense. Okay, so when you get to part B, again, you want to make sure that you are writing in complete sentences. You want to make sure that you are clear and to the point. You do not need to restate the question, but you can say something straightforward and simple. So describe the effect of caffeine on each of the short-term and the long-term memory of the bees. So if I'm typing something in, you could say something like, caffeine does not affect the short-term memory in the bees. However, caffeine, oops, you guys get to see my spelling mistakes. Improves the long-term memory as seen in the increased probability, and this is just taking too long to type, ability of revisiting nectar source after 24 hours. All right, so if I go back and correct my spelling mistakes, this should be pretty straightforward. And you would get 2 points for that by identifying both the short-term memory and the long-term memory of each section. Now, as you're going through this particular FRQ, it's not going to tell you necessarily where the points are going to come from on each of the subsections. It will tell you that overall you will get 10 points on this particular question, but it's a good idea to do your best to answer each subsection as best you can. Some subsections, as you can see, are worth more than others. The graph itself was worth 3 points, but this little part right here was worth 2, and then there's other parts that are worth 1. So be sure that as you are answering this question, you're thinking about trying to get the most out of each question, because you may, like I said, get an easy point here or there. All right, so remember on each of these two, we are looking for the command term, and we don't need to do anything beyond what that bolded word says. So we don't need to add any additional information. You don't get extra points for being extra explanatory. So make sure you answer what the question is asking. All right, so this is the real one I wanted to spend some time on tonight. Design an experiment. So it's going to ask you here to design an experiment using artificial flowers to investigate the potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits by bees. Identify the null hypothesis and an appropriate control treatment and the predicted results that could be used to reject the null hypothesis. So this is actually asking a lot in here. And this question itself, this sub-question, is going to be worth 3 points. So it's a good chunk of this overall FRQ. So within this experimental question, remember that you are going to try to identify those things we talked about before. So you want to identify your variables, probably. In the long run, on the scoring guidelines, you don't get specific points for those variables, but you should be describing that experiment and those variables as best you can, because you could get points for it on a different scoring guideline set. It just depends on what they're looking for within the question. All right, so let's be as brief as possible, and I'm going to try to type as quickly as I can with a potential response. You guys can start keeping your own answers or correcting as you go, but think about how you would write out, or how you get started writing this FRQ or this particular experimental part. So again, you don't really need a topic sentence, but if you want to get started, you can say something like to conduct an experiment to test the potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits by bees. Make sure my spelling is, oh my goodness, where'd it go? Let's see, researchers could use a control group of what? So you want to think of your control group, because that's part of something that you're supposed to identify. All right, my screen is going slow, I think, because I'm running the screencast at the same time. So my typing is like 2 seconds behind my fingers. All right, so go ahead and think really quick. If you have a control group in mind, go ahead and put it in the chat and we'll see what you guys have to say. Okay, great. I'm seeing a lot of suggestions of flowers without caffeine. So that's exactly what we're looking for. Artificial flowers with no caffeine compared with our experimental groups of artificial flowers with increasing caffeine. So I'm going to go ahead and I'm just going to dump in what I wrote before, because I did sort of a practice run, and then we're going to add to it. Okay, so it could use a control group of artificial flowers with no caffeine compared with experimental groups of artificial flowers with increasing caffeine concentrations. Let's just say from 0.1 to 0.5. All right. We could also say, maybe if you wanted to be specific, you could say paired with the nectar, the artificial flowers. All right. So again, you don't have to like make different paragraphs. I'm just making a paragraph break to make it easier for you guys to see. thinking about the dependent variable, you're going to measure the number of bee visits over a 24-hour period. So you would state that by just saying the dependent variable. And then what would be our null hypothesis? So we have to state a null hypothesis. It asks you to do that. And it's a good strategy to do that in any experimental question as well. But think about what you could say. So if you have a null hypothesis idea, go ahead and throw it in the chat for me real quick. Oh, good question in the chat as well. Do we need a positive control as well as a negative control? Good enough. In this state, in this particular type of question, a negative control is good enough. Great. So I'm seeing some caffeine has no effect on the memory of bees. Caffeine will not have a, so you don't want to use a double negative, but yeah, okay, good. So caffeine will not have a negative effect on bees. Caffeine has no significant effect on the memory of bees. Excellent, guys. Okay. So I'm going to throw in the one I wrote, but a lot of you guys are saying great stuff. The null hypothesis would be that increasing caffeine concentration has no effect on the number of floral visits by bees. So I was specific to relate it exactly back to my dependent variable that I'm measuring here. And then the question asks the predicted results that could be used to reject the null hypothesis. So this is a little tricky, but it's asking to say, what would this experiment show, or what would we observe in this experiment if this null hypothesis were to be rejected? And so that could be something, I'm just going to paste my answer to save some time. In order to reject this null hypothesis, the experiment would have to show results where the number of floral visits by bees is significantly different at increasing caffeine concentrations. So the predicted results for this experiment that would reject the null hypothesis would be that the highest caffeine concentration yields the fewest bee visits. So I threw in a hypothesis there. Wasn't it really in a great statement, but it was one way to show that this is what could happen in these results if the null hypothesis is rejected. And remember, that is showing that there is some sort of effect of the experimental treatment. Okay, so we're going to move forward with the rest of this question, but that is all that experimental question needs. Null hypothesis, control treatment, and predicted results. You could answer this question probably a little bit simpler than what I did right here, but for the full design and experiment part, it's really a good idea to identify your variables and how you would set that up. Again, I didn't say like, I would go to the store, I would buy caffeine, I would do all these crazy things. It's really just like how you would set up the different incremental treatment groups. And then, and I didn't even list all my different treatment groups. I just said increasing caffeine concentrations from here to here. You could even do it up to like, I don't know, 0.9 or something. It's up to you as long as it shows that you understand that we are showing an increased caffeine concentration, and that's what we're testing. All right, because we're running a little bit low on time, I'm going to go through and finish this B question, and then we're going to go through that next one that I have listed, and then I want to save some time for questions to answer at the very end, and then we're going to wrap it up for tonight. All right, so please keep putting in questions in the Ask a Question section of the Crowdcast, and we're going to go through this last part here. All right, so here at the very end, it's asking you to propose one benefit to plants for a proposed plant benefit to produce nectar with caffeine and a lower sugar content, and then propose one cost to bees that visit the flower plants that produce nectar with caffeine and a lower sugar content. So this one's kind of open-ended. It's a little bit less clear what exactly they're looking for. But if we're thinking like evolutionarily, or we're thinking at the organismal level, a benefit to plants with caffeinated nectar and lower sugar content would probably be that there'd be more pollen distributed by increased visits. So the more caffeine there is, the more the bees are coming back, and then the more they're getting pollinated. And they can save the energy for other uses, so that they can maybe save that for more metabolic processes like growth or producing other hormones or whatever they need to do. So one cost to bees on the other end, if you think about what they would typically use that sugar for, if the sugar content is lowered and the caffeine is increased, maybe they aren't able to bring that sugar or that nectar with lots of sugar back to the colony. Maybe they're not able to produce good quality honey, and then that's going to be an energetic cost for the colony. So you could say something like, one cost to bees visiting flowers with nectar containing caffeine and lower sugar content would be that the colony may produce a lower quality honey with less energetic value. So when you think about these types of questions, again, I'm only saying one cost and one benefit here. I'm not listing a bunch. I'm not putting it in bullet points. I'm still putting it in complete sentences. This bracket thing here is not a complete sentence. I'm going to fix that. Plants could save the energy for other uses. There we go. I'm not very specific in here, but I don't need to go into a great deal of detail. Okay, moving forward, I wanted to hit this last question here, the 2013 question number 1. So if you guys want to go ahead and click on that link and pull it up. This is a choice chamber question. So we have an experimental setup. You might have done an experiment similar to this in your biology classes. A lot of times, though, you will see experimental designs that you've never heard of before, and that is okay. Again, like we said before, these questions 1 and questions 2 are meant to trip you up. It's meant to show you something new, but to test your scientific skills and see how you can apply all the skills that you learned in AP Biology over the course of the year to a new situation. So don't get freaked out by that. All right, so I'm going to give you guys a second to read through 2013 question 1 while I take a look at some of the questions that have popped up here. All right, all right. People want to go over making a cladogram. I can talk briefly about that towards the end. I don't have a good example pulled up for you guys, and a lot of times that involves, having a drawing ready to go. But there are some great questions involved making cladograms. I'm going to pull up that slide with all of the FRQs from previous years, and I'll show you where you can go, where you can find to make a cladogram. So we will get to that towards the end, but I will make sure I show you guys where to see those past cladogram questions. All right, another question from here that I'm answering while you guys are taking a look at that 2013 FRQ. Is there any FRQ questions about math that's not Hardy-Weinberg? Yes, absolutely. So I have seen FRQ questions where you're asked to calculate a density of a population. There are questions in FRQs where you're asked to calculate rates. There are questions in FRQs with math with chi-square standard deviation. I would say Hardy-Weinberg does not show up in the FRQs as much as you would think, even though some teachers really like to practice it a lot. You're probably going to see a Hardy-Weinberg a little bit more in the multiple choice sections, maybe in the grid-ins, so keep an eye out for that. But yes, there are FRQ questions with math that are not Hardy-Weinberg. That is a good question. All right. So let's get back to the long FRQ, and then from there, we're going to try to wrap this up in the next 10 or so minutes so that I can let you guys go and study for your other APs. Okay, so within this FRQ, if you guys can still see my screen, great. All right, we're still there. Perfect. You are shown an experiment where you have a choice chamber and you have flies. And on one side of the chamber, you're going to be able to see the setup where there's a glucose-soaked cotton ball, and then the other side there is a dry cotton ball. And in this experiment, the positions of the flies are observed at different times, and the experimenters are going to calculate data to see if there's any sort of taxis or attraction to one particular side of the chamber or another. So you are asked at the very beginning to predict the distribution of flies in the chamber after 10 minutes. So if you think about making a prediction here, they actually do not have a specific prediction that you must make. You just have to be able to justify it correctly. Obviously, most students are going to predict that there will be more flies over towards the glucose after 10 minutes. But the justification you could write for that would be something like the flies are maybe attracted to the glucose because it's representative of food source for them. So after 10 minutes, there'll be more flies in the chamber with the glucose-soaked cotton because glucose provides energy for the flies, the food source, and it will attract more. You could go into a little bit more detail, but like I said, save your time on your FRQs. I didn't need to write anything beyond what I just put up on the screen. You do not need to make this long-winded or wordy or restate the experiment or say like, if a scientist set up a choice chamber and put glucose on one side and dry cotton on the other, you can leave all of that out. It really needs to be just an answer to the question. So it's one prediction. I have it in one sentence here, and it's not even the most grammatically beautiful sentence, but it answers the question. All right. If we're looking at this experiment and proposing one specific improvement to each of the following parts of the experiment and then explaining how it will affect the experiment, oops, I got one of the old questions in here. There we go. This part is actually potentially 4 points. So for each of the controls, you would get a point for answering an improvement. Excuse me, for the experimental control, you would get a point for telling what the improvement would be and then explaining how or why. And then you would get another point for identifying an improved environmental factor you could introduce and then explaining how or why. So there's a lot of possible ways you could improve this experiment. One of the most obvious ones is to improve the experimental control. You're probably going to want to make that dry cotton soaked with water as well, because you want to make sure that the glucose is actually what's attracting the fly, not the texture or the moisture. So that's all you really need to say about the first one, and that's a good enough explanation. So the proposal is right here. And you notice how I didn't even separate those two parts, the proposal and the explanation. It's all together in one sentence. So to improve the experimental control, the researchers could exchange the dry cotton for the water-soaked cotton. The explanation is here, which would ensure glucose is what attracts the fly, not the moisture or the texture. It's all combined in one. You don't have to say, like, I propose this. This is my explanation. Again, you can condense it and make it pretty simple. Environmental factors, you could list a bunch of different things. You could say, like, maybe you would do this experiment over and use different concentrations of glucose and showing increasing incremental treatments. Maybe you would say you would use different temperatures. Maybe you would say you would want to add variation to the experiment about the time of day just to make sure that it's repeated lots and lots. But anything that you do, just make sure you add that explanation about how it's going to attribute the movement of the flies only to the variable you are controlling for. So if you're doing this at different times of day, you're ensuring that those flies are not just going over to the glucose because it's on the sunny side of the room. If you're doing this in different temperatures, you are ensuring that the flies are not just going to the glucose because that cotton ball is a little bit warmer and a little bit cooler. So for example, to improve the environmental factors, the researchers could use incremental concentrations of glucose, which would attribute the movement of flies only to the glucose preference. And hopefully you would see an increased amount of flies as the concentrations of glucose increased, similar to our bees and caffeine experiment. All right. Finally, to the chi-square test. So I've seen in the chat some of you guys are a little worried about chi-square. Some of you guys have been doing it all year long. Chi-square is a statistical test. It is really not that scary, but I want to make sure you are familiar with how you could set this up and what it would look like if it does show up on your FRQ. So it tells you to perform a chi-square test on the data. And it's going to give you a very specific way it wants you to do it. It gives you a table on this question. So you actually have to fill out the table to get this right. I know some of you like to do chi-square in your head or in the scratch, like the sidebar of your notes. But you do need to fill out the table if one shows up. If there's no table, you don't need to create one. It's always a good idea to sort of lay it out like this. When you are looking on your formula sheet, I'm going to scroll down here. This is the section of the formula sheet where the chi-square formula is going to be. Chi is this symbol here, and then we call it chi-square because we square that value. There's no actual squaring of the value. So even though it's called chi-square and there's like an exponent with a 2 here, once you calculate the sum using this sigma value, you do not need to square anything. So keep that in mind. Some students get that confused. This symbol here is a sigma. This means sum. So what we're going to do is use this formula here for each of the values in our table, and then we're going to add them all up, and that's our total. So if you've not practiced this before, that's what this means. The O's and E's are pretty straightforward. It's expected and observed. And it actually breaks it down for you right here. So within this table, and you guys can kind of fill this in with me, we are given the, this is the actual data table from the FRQ that I have identified, or I just screenshotted from the actual FRQ. Our null hypothesis I've already written in. So I said, if we're creating a null hypothesis for this experiment, we're going to say that there's no effect. So we could say the flies are going to be evenly distributed across the 3 parts of the choice chamber. And that there's no really difference between any particular part of the chamber. I forgot to mention here, this is now identifying a new sort of experiment based on the first part. So it is a good idea to jump around from FRQ to FRQ, but you may not want to go out of order of the questions in the subsections of the long FRQs, because sometimes they build on each other. And when you get to part C in this question, what it's telling you is that there's a new experimental design where scientists have changed it up a little bit, and we have a different situation going on. So keep that in mind when you're moving around. All right, so when we're filling out this table here, for our observed values, and I'm gonna make sure I can type in here, this is given to us on the chart above. So this is a table. I made this in this doc, but on the actual FRQ, the table was provided for me, and this is what you have to fill out. So make sure if there's a table, like I said before, you are filling it out on the actual FRQ. So for the end with the ripe banana, that is given to us. And this is the end of the experiment, so 10 minutes. And we saw the end with the ripe banana is going to be 45. The end with the unripe banana is going to be 12. I'm skipping around, sorry about that. And then the middle is going to be 3 flies. If we were to total all these flies up, oh, my table cut off, it would be 60 flies total. So what we know is that we have to evenly distribute all of our 60 flies across the entire experiment. Let me make this smaller. Perfect. Okay. So if our null hypothesis is that the flies are going to be evenly distributed across the 3 different parts of the choice chamber, then each one would have an equal amount of flies in it. So the end with the ripe banana would be 20. The end with the middle banana would be 20, and then the end with the unripe banana would also be 20 flies. All right, so time to actually do our calculations. We're going to go through and we're going to take our expected value and subtract, take our observed value and subtract the expected value. So observed minus expected, square it, and then divide it by the expected. I'll let you guys practice out the math. I'm going to give you the values right here really quickly just to save us some time. And again, just kind of straightforward algebra. We were taking one, subtracting another, squaring it, and dividing it by the expected value. Now we're going to add all these up. This is this part of the equation. So once we did observed minus expected, I'm getting it backwards, guys, squaring it, divided by expected, we're going to get a value. That is this. We're going to do it for this value and this value. And then we add all of these up, and it should get you 48.9. This is the chi-square. You do not need to square it. Just leave it. So what do we do now? What does this value tell us? We're now going to have to use this table with our degrees of freedom, and we're going to have to explain whether or not our hypothesis is justified, our null hypothesis is supported or rejected by the chi-square test. So to answer part C, we are actually done. We created a null hypothesis, and we performed a test with the data. We got our values. The actual part where we have to explain is in part D. So you get points for doing this table and entering a null hypothesis, and then you get points for an explanation. So it's kind of nice that they split it up, because if you screw up one part here, maybe you can still get a point here, or you get a point for your null hypothesis, even if you mess up some of your calculations. So again, if you're not familiar with this, it's okay to try, and it's really okay to take a stab at it, even if you're not sure if you're going to get it correct. Okay, within any sort of data set, the number of categories minus 1 is going to be your degrees of freedom. So in this case, we have 1, 2, 3 different categories, 3 different conditions where these flies could be. We subtract 1, and that is our degrees of freedom. So in this case, we have 2 degrees of freedom. Now we're going to focus on the p-value in the chart that matches with our degrees of freedom and our value that is going to equal a 95% confidence interval, which is p equals 0.05, which is commonly used in biology. So what this will give us is, if we look at our chart for 2 degrees of freedom down to 0.05 here, that's going to be a critical value of, and you guys probably are ahead of me and know this already, 5.99. So now we have this number. Can we reject our null hypothesis? We have to go back up to this value here. If this value is greater than this value, we are going to reject our null hypothesis. So I'm going to write it down right here just so I don't go back and forth on the screen. And yes, 48.9 is definitely greater than 5.99. So this null hypothesis is rejected, and that means there is an experimental significance. So any sort of explanation we could write out would look something like this. I was a little wordy here, but you guys can kind of take my example and run with it. So with the 3 categories for the flies to select from, 2 degrees of freedom are used. You guys notice I wrote out all of this up here. this is technically not part of my answer. This was just scratch notes for me. Remember, I have to write in complete sentences. With the 3 categories of flies to select from, 2 degrees of freedom are used. When using a p-value of 0.05, the calculated chi-square value of 48.9 rejects the null hypothesis in favor of the alternative hypothesis. We did not state an alternative hypothesis, and that is the idea that there is going to be some sort of effect from this experiment. You can just mention it. You don't have to state it. It's a good idea to state it too, though. Glucose does have an effect on fruit fly behavior. That's our alternative hypothesis. Sorry, I did state it. And as it is greater than the maximum value in the table of 5.99. All right. I left part E on here. I already filled it out. This is a proposal of a model of how environmental cues affect the behavior of the flies. This is just like, is there a stimulus? Is there a response? What's going on here? I want to take a few more minutes to answer some of the questions that popped up in the question box. So if you guys have any last minute ones, please feel free to enter those and upvote any ones that you think might be interesting. While I am doing that, I'm going to also pull up, and this hopefully will be pretty easy. I'm going to pull up that chart so that you guys can see all of the old FRQ sections. Let's see. Here we go. Perfect. Okay. So we are back in action. Yes. Okay. So these, again, are the questions I've been mentioning. These are the ones you guys can look back at in the past. Okay, I'm going to look in the chat really quick. So Lori says, will the P we need to look at be given to us, like whether it's 0.05 or 0.01? Yeah, and Daniel's correct. In my experience, it's always been 0.05. 95% confidence interval is like the conventional confidence interval we use in biology. Unless they state otherwise, go with 0.05. Always use that one. I'm glad chi-square makes so much more sense. Okay. Yeah. We're always using 0.05. Okay, great. All right, questions real quick, and then we're going to wrap it up for the evening. Thank you guys so much for sticking with me, and I appreciate all the questions that you are asking. So what are some of the things that are guaranteed on the AP exams? Like one free response about labs. Okay, so guaranteed you are going to have to use your science practices. So these are things like analyzing data and interpreting models. It's really hard to say exactly what's going to show up. A lot of the questions that they generate are based on things that are published in research and that you probably haven't seen. These might be things that are new. Most of the FRQs are generated from actual research papers. So you can go back and find all the experiments that these are based on after they're released to the general public, which is interesting. But that means you are going to see things that you have never seen before. But being aware of your science practices, like being able to interpret data, being able to interpret a model, being able to use a cladogram to interpret evolutionary relationships. All of these things you should be able to apply even if you're seeing the information for the first time. So keep that in mind. All right. So I keep clicking these questions and forgetting to click the answer things. So I'm not going to go over Hardy-Weinberg tonight, but there are a lot of great Hardy-Weinberg resources. I think Fiveable has a past review on Hardy-Weinberg. I think you can go to my YouTube channel and find some Hardy-Weinberg thing as well. So null hypotheses, again, I'm going to go over that real quick one more time. A null hypothesis is the idea within an experiment that the treatment or whatever independent variable you are putting in the experiment is going to have no effect on the actual experimental results. And the null hypothesis is a way to use your experimental data and apply statistical significance tests to it. So we use it in the case of statistics, and it's really common in scientific experiments to create a null hypothesis that is very simple, like the treatment will have no effect on whatever we're testing. So there's a lot of good null hypotheses practice sheets that are out there, but just get in your head null hypothesis, think like no effect, there'll be no treatment. All right. Oh, thanks, Mahmood. Thanks for joining in. All right. I'm going to answer this last one, and then I'm going to sign off for the evening. Can we answer the parts of the FRQ out of order, like B, A, C, A, B, C? Okay. So like I said, I don't recommend tackling the questions out of order, but you can answer them out of order as long as you label them. So if you want to put like, well, you can answer them out of order, and if it's as long as it's within the same section. The readers will read the whole thing. It might be harder for them to identify which parts you're answering. So I always recommend, if you're starting with whatever part, if you're starting with A or starting with C, to put that little letter in parentheses before you start your paragraph, and that'll help the readers find which answer you're responding to faster. Again, it's not going to be the end of the world if you do it out of order. It's not going to be the end of the world if you don't label each of your responses. It's just a good strategy. Okay, yeah. And so I have people asking if I'm going to put my responses in the chat at the end. I will absolutely do that. Thank you guys for sticking around so long. I am really excited to have done this with you and all the people who joined in. I'm going to leave the link to these slides with the Fiveable people, so you will be able to access them if you have access to that. And then the link to my responses will be in the chat at the end. So let me share that with you, and then we are about to sign off. So thank you guys so much for joining us. And I hope you have a good night and enjoy studying for the AP Biology exam. You've got a little bit of time left, and I know you guys are going to be able to do great on this. Thank you guys so much.